1, 2, 3, yeah.
The first part of mitotic phases in animal cells is interphase. In interphase, cells have a distinct nucleus and, in the G2 stage, 2 pairs of centrioles. The chromosomes are uncoiled and are visible only as chromatin granules. These are the chromosomes. The second phase is prophase. During prophase, the nuclear membrane and nucleolus disappear. The replicated chromosomes coil tightly to appear as rod-shaped structures. Each pair of centrioles migrates to opposite ends of the cell. And spindle fibers extend between the pairs of centrioles. Each pair of centrioles and its radiating astral rays constitute an aster at each end of the football-shaped spindle. In late prophase, spindle fibers from opposite poles attach to the centromeres of the sister chromatids. The third phase is metaphase. In this brief phase, it is characterized by the chromosomes lining up at the equator of the spindle. It is the lengthening and shortening of the spindle fibers that move the chromosomes to the equator of the spindle. The next phase is anaphase. Anaphase begins with the separation of the centromeres of the sister chromatids. Then, the shortening of the spindle fibers pulls the sister chromatids toward opposite poles. Once the sister chromatids separate and begin moving toward opposite poles of the spindle, they are called daughter chromosomes. Thus, a cell in anaphase contains 2 complete sets of chromosomes. The next phase is telophase. In telophase, a new nuclear membrane forms around each set of chromosomes to form 2 new nuclei. The nucleolus reappears and the chromosomes start to uncoil. Usually, cytokinesis, cytoplasmic division, begins prior to the end of telophase. A cleavage furrow appears, which continues to construct until the parent cell divides to produce 2 daughter cells. And here are the 2 daughter cells. Go.
So I'll be talking about meiosis 1. And to start off with, we have prophase 1, which is 2 sister chromatids joined together, and they join together side by side in a pairing called synapsis. And just a fun little fact, tetrads are 4 chromosomes found in synapsis, but we're not going to talk about that. So the chromosomes coil together to appear rod-shaped, and the nuclear membrane disappear and the spindle forms. It's very vague, so you're just going to have to go on that. Okay, then we move on to metaphase 1, which is synapse chromosomes lining up where they attach to spindle fibers. Okay, so yeah, you see those 2 there, and they're lining up and all that good stuff. And then we go on to anaphase 1, which is separation of the members of each chromosome pair. And the centromeres don't separate, but they're just polarized at each side. Okay, and then finally we have telophase 1, which is nuclear membrane is formed and arranged in each set of chromosomes. The nucleus reappears, and then cytokinesis finally separates the parent cell into 2 daughter cells, and the nucleus of the daughter cell contains only 1 member of each chromosome pair. Keep that in mind. There you go.
Yeah.
Okay, so I'll be talking about crossing over. So crossing over happens during meiosis. I've kept 2 of Annie's pictures here. So it happens between prophase 1 and metaphase 1. It occurs in the first division of meiosis. During this process, chromosomes pair up with each other and exchange different segments of their genetic material to form other reactive chromosomes. So crossing over usually occurs when matching areas on matching chromosomes kind of reconnect, and then they make another chromosome. So it's a result of recombinant chromosomes during sexual reproduction. Yeah.
So I'm going to be talking about meiosis 2. So we start with prophase 2, and it's the loss of the nuclear membrane and the rod-shaped chromosomes appear. Then in metaphase 2, the chromosomes line up in the middle, and each chromosome has 2 sister chromatids joined by centromeres. And in anaphase 2, there's the separation of the centromeres, and then the new daughter cell or daughter chromosomes move toward opposite ends. And then in telophase 2, new nuclei forms, and 2 haploid daughter cells form during cytokinesis.
Okay, so when talking about mitosis and meiosis, it's kind of easy to get them confused, not just because of the name, but because they kind of have similar functions or they have something to do with one another. But the main difference between them, meiosis has 2 rounds of genetic separation and cellular division, while mitosis only has 1 of each. In meiosis,
Homologous.
chromosomes separate, leading to daughter cells that are not genetically identical, but in mitosis, the daughter cells are identical to the parent as well as to each other. Mitosis only has 1 of each. In meiosis, during this process, oh my god, start over.
No, we're still going. You gotta just
What is
finish it.